everyone. In today's video, we are going to learn about Griffith's experiment and the transforming principle. This classic experiment was a stepping stone towards the eventual discovery of the genetic material that we today know as the DNA. Before learning about his famous experiment, let us know a bit about the man himself. Who was Griffith? Frederick Griffith was born in England in 1877 and went on to study medicine at the University of Liverpool. Later, as a bacteriologist, he was studying bacteria that cause pneumonia in humans. These bacteria are commonly referred as pneumococci and they infect the respiratory system in hosts, eventually leading to pneumonia. While doing these experiments, Griffith's goal was to develop a vaccine against pneumonia. The bacteria Streptococcus pneumoniae comes in various strains or types. Griffith, in his experiments, worked with two different strains of bacteria. That is, the smooth strain, also known as the strain S or type 3S, and the rough strain, also known as strain R or type 2R. Both these strains differed in appearance and virulence. The S strain bacteria produce smooth colonies on a culture plate. This is due to the presence of a smooth capsule surrounding the bacteria. The capsule is nothing but a protective layer of polysaccharides that protects these bacteria from the immune system of the host. This property makes the S-type bacteria highly virulent and pathogenic. The R strain bacteria lack the protective capsule and therefore appear as rough colonies on a culture plate. They can be killed by the immune system of the host and are therefore harmless. While looking for a cure for pneumonia, Griffith injected mice with both the S and the R strains of bacteria to test whether they infect and cause pneumonia. In the first set of experiment, live S strain bacteria were injected in mice. As expected, the mice developed symptoms of pneumonia and died within few days. In the second set of experiment, Griffith injected mice with live R strain bacteria. The mice could survive against these bacteria and showed no symptoms of disease. In the third set of experiment, the S strain bacteria was subjected to high temperatures and killed. Heat-killed S strain bacteria were injected in mice and as expected, the mice could survive because the dead S strain bacteria were not capable of causing the infection. However, the experiment took an interesting turn on the fourth set when Griffith injected a mixture of heat-killed S bacteria and live R bacteria in mice. The mice developed pneumonia and eventually died within a few days. Individually, both the heat-killed S bacteria and the live R bacteria are not deadly. However, when injected together, how could they lead to pneumonia in mice was puzzling. Analysis of samples from the dead animals revealed the presence of both live S strain and live R strain bacteria. Griffith concluded that the R strain bacteria have taken up some element from the heat-killed S strain bacteria, which has now converted them into virulent S strain bacteria. In other words, the R strain bacteria have been transformed into the deadly S strain bacteria. Griffith named this process as the transforming principle. This simple experiment proved that organisms can acquire new factors from other organisms and their surrounding environment, and that non-heritable exchange of genetic material, that is, horizontal transfer, is a regular phenomenon in nature. Griffith, however, could not explain what exactly this transforming principle was. This puzzle was solved 16 years later by another group of scientists. Avery, MacLeod and McCarty showed that the transforming principle in Griffith's experiment is the DNA.